We're going to go ahead and get into chapter 12, the DNA section. Even though we haven't taken a test over the genetics, I will incorporate some genetics in the DNA section because they are directly related. As we know from the genetics section that all of our traits are located on chromosomes, and our chromosomes are made up of the chemical factor called genes. And what DNA does, it's going to code and produce proteins to make those genes and express those genes. The picture I have there is a model of a DNA. It's called a double helix, and that is because it's two strands that are connected and then they are twisted to make the double helix shape. Today our main focus is going to be on chapter 12, section 1. We're going to talk about the scientists that contributed to understanding that DNA was responsible for passing on these traits and what the structure is. And then the last part we will actually talk about the structure of the DNA. So we start out with Griffith and what he called transformation. He was a British scientist and he started using bacteria that caused pneumonia. When you're looking at the bacteria that cause pneumonia, you have the disease-causing, which is a smooth colony, and you have a harmless, which is going to be the rough colony. So what he did was he took mice, and the first one, he took the mouse and injected it with the disease-causing bacteria, and the mouse developed pneumonia and died. The second one, they were injected with the harmless bacteria, therefore they did not get sick. Now the third, he took the disease-causing bacteria and he heat killed it. So therefore when he injected the mouse with it, then it ended up living because the bacteria was actually dead. Now here's the biggest one. And the fourth one, he took the disease-killed bacteria and combined it with the harmless bacteria, but it was still alive. When he injected the mouse still died. Now what he decided is that the heat-killed bacteria was able to pass some genetic information to the harmless strain that was alive. Therefore it died. What this was called transformation. That the harmless strain received information from the heat-killed and then therefore made it the disease-causing bacteria again. Like I said, the main point of this was to show that genetic information was passed on. So we knew there was something in our cells that will pass on some of this genetic information. Now they had to figure out how this happened. This leads us to Avery. And he also took Griffith's work, but he wanted to decide which molecule was important for this transformation. Now he was able to take enzymes, and he was going to destroy different things. Now if you recall from chapter 2, we have proteins, lipids, carbohydrates, and then other molecules. And we are going to talk about the RNA later on, but what they did was they killed all these proteins, lipids, carbohydrates. Now, the transformation still occurred after he killed off these things. So obviously, these were not the organisms that were responsible for passing on our traits. Avery and the other scientists, they continued to repeat this. And when they destroyed the DNA, transformation did not occur. This is how they finally decided that DNA must be the factor that was transferring the traits. So a key concept here is Avery and the other scientists discovered that the nucleic acid, DNA, stores and transmits the genetic information from one organism to the next. All right, so that is enough about these scientists, kind of the main points of how they figured this out. Now we're going to talk about the main structure of DNA. As we saw, DNA is a very, very, very long molecule. Now what it is, it's made up of many smaller parts. So DNA is known as a polymer. Poly, which is right here, means many. So when we talk about DNA, it is a polymer because it's made up of many smaller things called monomers. Mono means one. So it's kind of like a puzzle. You have one individual puzzle piece, and you put all these individual puzzle pieces together, and you get what's called a polymer, and that would be DNA. In DNA, a nucleotide is a monomer. So the nucleotide is the individual small piece that makes up the larger piece of DNA. So like it says here, mono is one piece of a polymer. When we're looking at a DNA molecule, DNA is your polymer and nucleotides are your monomer. Therefore, DNA is made up of many nucleotides. Now when we're looking at a DNA strand, here's a DNA strand. One nucleotide will be a deoxyribose sugar, there in the blue, a phosphate, which is the circle, and in the middle you have what are called nitrogen bases, and that one is the A, the T, the G, and the C. So right here, this is one nucleotide, and then it's connected to the second nucleotide. The nitrogen bases are really what code for our genetic makeup, which is kind of weird because there's only 4 nitrogen bases, but the order of these nitrogen bases on your DNA makes you look the way you are. You can either have adenine, guanine, cytosine, or thymine. Now they split those nucleotides into 2 sections. You have purines, which are your adenine and guanine, and you have what are called pyrimidines, which are cytosine and thymine. The reason this is important is you have this Chargaff's rule. He ended up discovering that the percentages of guanine and cytosine are always equal, and the percentages of adenine and thymine were equal. So he discovered that the purines were always about equal to the pyrimidines. So what this pretty much means is that when you're looking at a DNA molecule like this one, you have your phosphate group and your sugar and nitrogen base. Anytime you have the green adenine on one side of your DNA molecule, you have to have a red thymine on the other side of your DNA molecule. And this thymine is connected to the deoxyribose sugar and the phosphate. So once again, here's one nucleotide, and here's the second nucleotide. Now the next pair, you have guanine. Well, guanine always has to pair with the blue cytosine. Next one, thymine, pairs with adenine. So like we said in this, guanine always pairs with cytosine, and adenine always pairs with thymine. Alright, continuing to talk a little bit about the scientists. Rosalind Franklin, and then Watson and Crick were also important in this. They were the ones that actually found the DNA structure and what it looked like, and decided that it had 2 strands that were just held together. And that's where we come up with the double helix. Now the key concept down here is that using Franklin's X-ray, Watson and Crick decided it was called a double helix, and it had 2 strands that were wound around each other, and they were connected in the middle. So here's another picture of it. the two strands, and they're held together here in the middle by the nitrogen bases. Now, nitrogen bases have to be held together by something. Your nitrogen bases are held together by weak hydrogen bonds. So that's why you see these here in the middle, and those are those dots. So you have a nitrogen base here, a hydrogen bond, and a nitrogen base. And you always know that guanine pairs with cytosine. The last thing is that when we say guanine has to pair with cytosine and adenine has to pair with thymine, this is a rule called base pairing. So you know that adenine always pairs with thymine and cytosine always pairs with guanine. And it can be flip-flopped also. So when I say you can flip them, you can also have thymine on this side and adenine on the other side, or vice versa, guanine on this side and cytosine on this side. You just always have to make sure those nitrogen bases are pairing together. Please continue watching the next video for better explanation of the DNA structure and for directions on your assignment of building your DNA structure.